Welcome to Easy Microbiology.
Good morning, everyone. I am pleased to present to you our findings on Bordetella parapertussis, a bacterium closely related to the organism causing whooping cough. We will discuss this strain's characteristics, its occurrence within humans and other animals, and its growth media requirements. Paying attention is greatly appreciated. Bordetella parapertussis is a gram-negative bacterium closely related to Bordetella pertussis, the bacterium responsible for whooping cough. Unlike B. pertussis, it does not cause severe disease, but can still cause a milder respiratory infection in humans and sheep. These infections typically are located in the respiratory tract and need specialized media for culturing. Bordetella parapertussis is a gram-negative bacterium which can mainly be found in the respiratory tract of humans and sheep. It has a preference for the trachea and bronchi of its host, and requires specialized media for culturing. Bordetella parapertussis is a gram-negative bacterium related to the causative agent of whooping cough. It inhabits the respiratory tract and is usually cultivated on special media. B. parapertussis is similar to B. pertussis in the way that it is a tiny, non-motile cocobacillary bacterium with a capsule surrounding the cell. Bordetella parapertussis is a gram-negative bacterium which is primarily found in the respiratory tract of humans and sheep. The culturing of this bacterium requires specialized media, such as Bordet Gengu agar or Regan Low agar, supplemented with specific antibiotics. This media provides a suitable environment for the growth of Bordetella parapertussis. Furthermore, its cultural characteristics are similar to Bordetella pertussis, the causative agent of whooping cough. Bordetella parapertussis shares some biochemical characteristics with Bordetella pertussis, most notably being catalase positive and unable to ferment carbohydrates. However, there are certain differences in the metabolic profiles of the two species that should be explored further. For example, while B. pertussis is able to metabolize phenylalanine, B. parapertussis is not. In addition, B. parapertussis is unable to metabolize mannitol, arginine, glucose, and lactose, whereas B. pertussis can. Finally, B. parapertussis is able to metabolize maltose and trehalose, while B. pertussis cannot. Bordetella parapertussis primarily infects humans and sheep alike. It commonly resides in the respiratory system and requires specialized media for culturing. It's a milder form of respiratory illness than that of B. pertussis, although it can still cause discomfort. Fortunately, with the proper medical care, it's treatable. Bordetella parapertussis is a gram-negative bacterium related to Bordetella pertussis, the causative agent of whooping cough. B. parapertussis primarily infects humans and sheep, typically in the respiratory tract, and it requires specialized media for culturing. It produces some similar virulence factors to B. pertussis, including pertussis toxin and filamentous hemagglutinin, but in lower quantities. These virulence factors contribute to its ability to colonize and cause damage in the respiratory tract. Bordetella parapertussis, or B. parapertussis, exhibits various mechanisms to resist antibiotics, which are strikingly similar to those of Bordetella pertussis, the causative agent for whooping cough. These mechanisms include genetic mutations in target genes that encode antibiotic target proteins. However, such resistance is much less common in B. parapertussis as compared to B. pertussis, making it easier to treat infections caused by the former. Today we are discussing Bordetella parapertussis, a gram-negative bacterium related to the causative agent of whooping cough. Epidemiological studies have shown that this bacterium is responsible for a smaller number of cases compared to B. pertussis. It is also considered less contagious and less severe in its clinical manifestations. However, it is typically found in the respiratory tract and requires specialized media for culturing. Bordetella parapertussis is a gram-negative bacterium that primarily infects humans and sheep, and can be found in the respiratory tract. It requires specialized media for culturing. When it comes to pathogenesis, B. parapertussis attaches to the cilia of the airway epithelium, causing damage by releasing toxins and triggering an inflammatory response. While the clinical symptoms of B. parapertussis infection are generally milder than those caused by B. pertussis, it's important to take measures to prevent infection. Immunological responses to Bordetella parapertussis are similar to those elicited by Bordetella pertussis, or whooping cough. Both innate and adaptive responses are generated from the body's immune system. The primary form of protection against this bacteria is through the presence of antibodies, specifically immunoglobulin A, which reduces bacterial attachment and neutralizes toxins. Laboratory diagnosis of B. parapertussis infection is analogous to that of B. pertussis, typically involving collection of respiratory samples and their testing by means of techniques such as PCR or culture. It is important for these tests to be specific to both species in order to discern between them. To protect against Bordetella parapertussis, it is important to take preventive measures to reduce the spread of infection. While vaccines that protect against whooping cough, such as the DTaP vaccine, do not offer significant protection against this bacterium, good hygiene practices can help to reduce the risk of transmission. This includes washing your hands regularly and following respiratory etiquette. Infections caused by Bordetella parapertussis can be treated with antibiotics like macrolides or trimethoprim sulfamethoxazole to reduce the time and intensity of the disease. For cases of serious infection, antibiotics are very important to control the disease and lessen symptoms.
Responsible for a milder form of respiratory infection. Understanding its characteristics, epidemiology, pathogenesis, diagnosis, prevention, and treatment is vital for accurate diagnosis and appropriate management of infections caused by this bacterium. Similar to B. pertussis, it affects the respiratory tract, leading to respiratory infections. While laboratory testing is necessary for diagnosis, differentiating it from B. pertussis is crucial. Vaccination against pertussis may offer partial protection, and antibiotics can be effective in treating infections caused by Bordetella parapertussis. Early diagnosis plays a key role in optimizing management strategies for affected individuals.